Greeting to you all, wherever you are. This is our webinar series from International Academy of Cardiovascular Sciences. And we really make this platform to disseminate state-of-the-art science. Today is my distinct pleasure, honor, delight, and you name it, to introduce our speaker, Junichi Sadoshima. Is one of the most wonderful persons and one of the most brilliant and eminent cardiovascular and molecular biologists of our time. He is the professor and chairman, Department of Cell Biology and Molecular Medicine, executive director of Cardiovascular Research Institute at Rutgers University Medical School. I will sum up the first introductory paragraph and basically say Junichi is really internationally recognized, renowned cardiovascular scientist, molecular biologist, who has made numerous seminal contributions to several fields in medicine or in biology, including, I would say, he was the first in cardiovascular field to establish how cardiac myocyte can take mechanical signaling and convert it to a biological process such as expression of paracrine factor. These days, as you all know, paracrine factor is at the heart of everything we do. But Junichi was the first to show that you can, cardiac myocyte can convert mechanotransduction into mechanosignaling and expression of paracrine factor, specifically angiotensin II, and moved on, followed by that, showing that delineating several pathways involved in cardiac hypertrophic response. His landmark discovery in 1993 with Sergio Izumu was published in Cell and put Sadoshima in the forefront of cardiovascular sciences. Sadoshima is an MD Ph.D. Jun had a little bit of aberrant time in his life. He became a little off the road and became an interventional cardiologist. Can you believe Jun being an interventional cardiologist?
No.
Catheter to your coronaries, dilating arteries. Fortunately for us, he saw the light and he came from the dark side and joined the molecular biology and did a PhD and with Sergio Izumu at Harvard, and established his own laboratory in Michigan, and then was recruited to Rutgers, as I indicated, as chairman of cell biology and executive director of Cardiovascular Research Institute. As I mentioned earlier, his research really has been quite diverse in everything he has done. He has really done the state-of-the-art, top of the science work. His work on autophagy is basically first-rated in all aspects. His work on signaling pathway, he was the first one to study Hippo pathway in cardiovascular disease and establish in cardioprotective role of downstream molecule of Hippo in cardioprotection. He has been at the forefront of oxidative stress and oxidative signaling. He has been magnificent work in mitochondrial biology and mitophagy and sirtuins and so forth. He has published his work in numerous prestigious journals. He has over 400 publications, and I use the Clarivate, which is very strict criteria compared to Google. His H index is 109, over 54,000 citations. If you go to Google citation, you go over 100,000 citations, and he is always funded by NIH. Currently, he holds 4 R01 grants, and in addition to that, he's the North American director of one of my favorite research program, which is the Leduc Transatlantic Research Network of Excellence. Junichi is exceedingly active. He's a wonderful colleague. He is a distinguished chair professor, but you can ask him to review a paper that you think beneath him, but he is always volunteers his services. Is an editorial board of many journals, including JCI, Circulation Research, and JMCC and autophagy and American Journal of Physiology, to name a few. He is very selfless and dedicated and committed to academic work, never says no, which is really admirable. He has recognized for his scientific contribution to the field by numerous awards, including Katz Award from the American Heart Association many decades ago, but he decided to make that a pedigree. It's become a genetic trait, and all of his trainees that go through his lab, they get Katz Award. So therefore, everybody is volunteering to work in his lab because they will get a Katz Award. And he has received, in addition, Janice Pfeffer Distinguished Lecture Award, Thomas Smith Award from AHA, which is quite prestigious, from BCVS Distinguished Investigator Award, and AHA Merit Award. He is a member of ASCI and a fellow of AHA and International Society of Heart Failure Research. He is a great mentor, as I indicated. All of his mentor also great scientists, and I was just mentioning that there really has recruited a lot of good scientists in this program. They're wonderful people as well, and we really been very, very successful program in his department. With that, we are really delighted to host Junichi, my colleague DK. Dr. Agrawal couldn't be here today. On behalf of him, I welcome all of you to IACS. He runs all the program of IACS. Our panelists, I briefly mentioned that we have Jason Dyke in alphabetical order. He is Director of Cardiovascular Research Program at University of Alberta. We have Christoph Mack, who is the spokesperson on Comprehensive Heart Failure Center and Chair of Translational Research at University Clinic Wolfsburg. And he is also Chair of Coast Action at EU MetaHeart. And Professor Francesco Panini, who is Professor of Cardiology, Director of Translational and Experimental Cardiology, and Head of Cardiometabolic Division at University Heart Center, Switzerland. Gaetano Cenciulli, MD, Ph.D. He's Professor of Medicine at the City of New York. And finally, Dr. Tagmeyer, who has not joined us yet, but he's the Krebs. Oh, he has joined us. He is the Krebs of our time. He is the god in metabolism. And we are delighted to have him on board today. With that, I think we all complete. I turn the podium to Professor Junichi Sadoshima, which we're really, truly honored and delighted to host today. Junichi, it's all yours.
Okay. Do you hear me well? Yes.
Yes, sir.
Okay, great. Thank you very much, AJ, for a very kind introduction and also giving me this opportunity. And also, I'd like to thank all the panelists and also listeners. I really appreciate to come to this seminar. And today, I pick a little bit the brave subject that regarding metabolism. Our lab studies signaling pathway, but not a true expert of cardiac metabolism. So I really would like to use this opportunity to find out what's missing in our study. So I really appreciate if you can give us feedback. So today I would like to focus on molecular mechanism of cardiomyopathy and particularly the role of ATGL in lipotoxicity in obesity heart. And I focus on two signaling molecules. One is a GSK-beta. It's a kinase, and also FOXO1 is a transcription factor. And also during the study, I realized that I should study Western diet induced cardiomyopathy rather than high-fat diet. So I tell you the reason in the second part. And also since one of the my title includes GSK-beta, and this is a kinase that I've been working almost 30 years. And I started the study showing the GSK-3 beta is a very good inhibitor of cardiac hypertrophy, but it turned out it has so many different substrate, but its role is very different from cell type to cell type. So I recently generated conditional cardiac specific GSK-3 beta knock-in, so to study the role of GSK-3 beta in various cell type. So I show you this result at the end of my talk. So obesity, or as you know, the population is increasing dramatically, and it turned out that if you have body mass index more than 25, you have a higher risk to develop a cardiac disease, especially HFpEF. So studying obesity, how obesity affects metabolism and cardiac condition is very important. And my understanding of obesity condition is basically it developed two things. One is insulin resistance, and that actually decreased the glucose uptake and other glucose signaling. At the same time, it relies on fatty acid, especially if you have high fat intake, fat concentration increases, and obviously that uptake is increased, and fatty acid metabolism should be increased. And that causes oxidative stress and other inflammation and all kinds of mechanism to promote cardiomyopathy. So I have been working on this obesity cardiomyopathy through by studying mitochondria dysfunction, and particularly we're focusing on mitophagy mechanism. But since we have a good model, high fat diet, 60% high fat model, so I asked Takayama, this guy, to study what kind of signaling molecules are affected. And as expected, he found that AKT phosphorylation is decreased by fatty acid, the high fat diet. And this is most likely due to insulin resistance. And one of the important substrates is GSK-3 beta. And this actually GSK-3 beta phosphorylation is decreased. And this indicates that GSK-3 is activated. This GSK-3 phosphorylation usually inhibit kinase. So when it is dephosphorylated, it is activated. And also one most prominent changes in various metabolic pathway was the ATGL. This is a lipase hydrolyzed fatty acid triglyceride in cells, and this upregulation was prominent. So we wanted to study the connection among these signaling molecules. And ATGL is one of the rate limiting enzyme in which triglyceride is hydrolyzed. And this pathway is certainly important to produce fatty acid for oxidation. And also this fatty acid itself is able to activate gene transcription by binding to nuclear receptors. So nowadays, when you say something is upregulated, you are always asked which cell type. So we conducted single cell RNA sequencing, and we found that this fatty acid high fat diet increases ATGL. This is a gene name of ATGL, and also ABHD5. This is a activator of ATGL, so upregulated in cardiomyocyte fraction. So we so far we found that the high fat diet increases ATGL, and this actually supposed to suppress lipotoxicity and maintaining ATP production. And we thought that this mechanism is a very important mechanism, kind of an adaptive compensatory mechanism to maintain the cardiac condition in high-fat diet animals. So since GSK3 was dephosphorylated and activated, so it's nice, and also in cardiomyocyte. So it's nice to study what's going to happen. This mechanism is selectively abolished. So since our lab has been studying GSK many years, so we could easily develop cardiac specific GSK3 beta knockout mice. And we found that you see here, high fat diet induced upregulation of ATGL is abolished, you see here. And at the same time, we saw here is this one, this effect, the cardiac hypertrophy was enhanced, and diastolic dysfunction is increased, and also even LVEF is decreased, and also lipid accumulation is increased. So this fit the hypothesis that this upregulation is a adaptive compensatory mechanism, and without the GSK, ATGL is not upregulated, and all features of obesity cardiomyopathy was exacerbated. And so if this is true, maybe if you inhibit downregulate ATGL, it should mimic the condition of a GSK3 beta knockout. So we generated cardiac specific ATGL knockout mice, and then basically we saw the same thing as GSK3 beta knockout. So our hypothesis is that maybe I should move this. Our hypothesis is that high fat basically GSK activate GSK3 beta, and sorry, go back. And that upregulated ATGL and prevent cardiomyopathy. But if you inhibit GSK3 beta or ATGL, this actually this process is compensatory process is inhibited, and the cardiomyopathy is exacerbated. And I believe that this ATGL thing is consistent with what has been reported, including Dr. Dyke, other investigators. So the key question is whether ATGL is important. So we basically conducted a rescue experiment, and this is a myocyte culture. If you inhibit GSK3 beta, we saw more lipid accumulation, and if you overexpress ATGL in this condition, this lipid accumulation was rescued. And also ATGL downregulation did the same thing as GSK3 beta inhibition. And also we gave high-fat diet to mice, and sorry, I have to move this on again. Sorry, I don't know how to, oh, sorry. So we gave the ATGL AAV to GSK3 beta knockout mice, And we show that this diastolic dysfunction was improved. So we believe that this ATG downregulation is very important in mediating the effect of GSK3-beta knockout. So this clearly suggests the importance of ATGL gene regulation. So I was so curious how GSK3-beta connects to ATGL levels. So I looked at the literature and some people reported the FOXO1 could be the important transcription factor to regulate ATGL gene. So we conducted chip-seq of the FOXO1. Actually, we did this one for other purposes, but we proved out the data, and we found that the FOXO1 actually binds to the promoter of the ATGL genes in the heart. So we believe that the same thing happens in the heart as lipocyte. So we wanted to find the connection between GSK3-beta and the FOXO1, and if we're able to find this connection, we can explain how GSK3-beta activate ATGL. So we did a quick experiment to see whether GSK3 in cultured myocyte and see whether GSK3-beta can upregulate ATGL protein, as you see here. And then we repeated this experiment in the presence of FOXO1 knockdown, and we are able to suppress ATGL upregulation, as you see here. So we made a hypothesis that most likely GSK3-beta regulate the ATGL through FOXO1. But so far, we could not find a connection between these two. So we further investigated and see whether GSK3-beta can actually phosphorylate FOXO1. So basically, we did the in vitro, we generated FOXO1 recombinant protein and incubated with GSK3-beta. And we send it to proteomic core facilities. And they show me serine residue phosphorylate as I indicated here. There are multiple sites. But we found that maybe the serine phosphorylation 65, 465 and 467 is maybe interesting because it is located to LXXLL motif. This LXXLL motif is a known binding site of a SART1. So we made a hypothesis that if these serine residues are altered, it affects the structure of this motif, so that it may regulate binding of a SART1, and that SART1 causes deacetylation of FOXO1. And the reason why that we didn't focus at this time for other area was that because before this study, we recently showed that the SART1 actually protect the heart from obesity cardiomyopathy. And also we know that FOXO1 deacetylation does many good things, including activation of autophagy. So we are particularly interested in the role of this phosphorylation in these two sites. And actually, we generated phosphor-specific antibody, and we are able to show that the high-fat diet basically activate the GSK-3 beta and increases phosphorylation of these two sites. This phosphospecific antibody worked very, very well, and this accompanied by ATGL upregulation. And this actually, it turned out that this phosphorylation site was very interesting. And not only it is located near the LXXS LL motif, but also regulates translocation of FOXO1 into the nucleus. And most important regulator of FOXO1 is AKT. And probably many of you know when FOXO is phosphorylated by AKT, threonine 35 and serine, I forgot the exact number, 3 sites are phosphorylated by AKT. The FOXO go out from the nucleus. On the other hand, when AKT is inhibited, FOXO comes to the nucleus. And we found that this AKT inhibitor can induce the nuclear translocation of a FOXO, as you see here. But if you co-express this SA mutant, even this actually nuclear translocation is significantly inhibited. So we believe that this GSK3 mediated phosphorylation is more important than AKT-mediated phosphorylation, and both actually affect the nuclear translocation, but GSK regulation is stronger than AKT-mediated regulation. So to test the role of these two serine residues, we generated knock-in mice and then made a basically wild-type mice and a FOXO knock-in mice, and gave a normal diet and a high-fat diet, and then see what happened. And first of all, this double knock-in mice failed to upregulate ATGL. So this is same as a GSK3 beta knockout, and high-fat diet no longer upregulate the ATGL, as you see here. And we also conducted phosphospecific antibody, and we as expected, that the high-fat diet cannot induce phosphorylation in this site. So we believe that this phosphorylation site is very important for ATGL upregulation. And if you use the double knock-in mice, ATGL upregulation can be abolished. And if this upregulation is an important compensatory mechanism, lipotoxicity and cardiomyopathy should be exacerbated. And that was the case, and we saw a high-fat diet in double knock-in mice actually exacerbate diastolic dysfunction. This is echo parameters, and this is a PB loop. And also, even systolic function is a little bit decreased in this condition. And we look at the number of lipid droplet, and we saw a number of lipid droplet is increased by this knock-in. So we believe that this pathway is very, very important. And also, we just in case this double mutation have many various other effects. So to show that this actually exacerbation of obesity cardiomyopathy is mediated by the lack of ATGL upregulation, we supplemented ATGL, as you see here, and then these are able to improve diastolic dysfunction and also systolic dysfunction and hypertrophy was actually rescued. So we believe that this process is very important for ATGL upregulation and also inhibition of lipotoxic cardiomyopathy. And as I told you, that these two serines are next to LXXLL motif. So we thought that the third one might be involved. And as I told you that these are a previous study, but a gain of function mutation basically rescued diastolic dysfunction in the high-fat diet fed mice, and also ATGL level is higher. On the other hand, if you knock down SART1, the obesity cardiomyopathy was exacerbated. And we found that actually these sites are very important. And also when the SART1 binds to FOXO, the most important thing is level of acetylation decreases. So in the presence of GSK, we believe that GSK phosphorylate this site, and the SART1, FOXO1 can bind so that this one is a co-IP result, and the GSK3 and the FOXO1 interaction is increased, and FOXO1 acetylation level is decreased. So we believe that this phosphorylation allows two protein interact, and this causes deacetylation of lysine residues. And we look at the interaction between FOXO1 and SART1, and double mutation knock-in. If this serine residue is replaced with alanine, we saw interaction between FOXO and SART1 is decreased. So these are basically the summary of my first part of my talk. We believe that most likely GSK3-beta is activated by insulin resistance, and phosphorylate FOXO1 two sites near this SART1 binding motif, and that causes interaction of two proteins, and basically the FOXO is deacetylated and the ATGL transcription is increased. And we are so excited, and we submitted the grant, but grant reviewers are not so excited because one of the major reasons is that all you guys studied adaptive mechanism, and that's interesting, but may not explain disease. So not well accepted. So we thought that maybe we should find a condition in which GSK3-beta, this pathway is actually inhibited, not activated. The high-fat diet, these are activated as a compensatory mechanism, but we wanted to know whether any condition these pathways are downregulated. Then lucky, we found a paper published by Doug Rindowski, 2023. He said that ATGL is downregulated in response to Western diet. And the Western diet not only induce diastolic dysfunction, but also induce systolic dysfunction. So what is Western diet? And according to the literature, Western diet contains high fat, but high saturated fat, and also usually contains high carbohydrate, high sugar, and low fiber. And the consequence is the fact that it has a high fat is similar, but the consequence is one is HFpEF, but the other is a HFrEF. And I and AJ and others, Chris also just came back from Greece and Kalamata, and I enjoy the food, a lot of vegetables and fish. But at the same time, I found that this is AJ's comment. There are many obese people in Greece. And also my impression is there are many like sweet shop, so many cake and ice cream and chocolate shops. And I found more cake shop than Greek food shop in Kalamata. And I did the AI search, and AI also point out that the Greek people develop cardiovascular disease, and these become a serious problem in Greece. So Western diet is really looks like a Western diet is very bad. So I copied Doug Rindowski's protocol, and our Western diet composition is shown like this. And then basically we found the same thing as Doug Rindowski, and different from high-fat diet. We saw ATG has a tendency to be downregulated, and not only lipid droplet is accumulated, but also both diastolic and systolic dysfunction develop in our Western diet fed mouse model. And we are very excited to find that in high-fat diet, we saw decreases in AKT phosphorylation, but we saw increases in phosphorylation, increases in GSK phosphorylation, and also decreases in ATGL. So this AKT, GSK, ATGL connection are very different between Western diet and a high-fat diet. So, and then we wanted to know whether ATGL is a key. Actually, Doug Lindowski's paper also suggested that the ATGL is important. Understanding ATGL downregulation is important in understanding Western diet-induced cardiomyopathy. So we basically conducted this rescue experiment, and we basically expressed the ATGL in mice and then gave Western diet. And we no longer see decreases in ejection fraction, and also diastolic dysfunction was improved. And you see this pressure volume loop slope become more flat, so that this indicates that diastolic dysfunction is improved. So to examine whether this actually causes FOXO1, 465, 467 dephosphorylation. I mean, when AKT is activated, GSK is inhibited, so that the dysphosphorylation site is not phosphorylated. So we generated a cardiac-specific FOXO1 double knock-in mice, and we examine whether this dephosphorylation alone can mimic the condition of a Western diet. And this is a scheme how we generated the cardiac specific knock-in. And we saw basically these mice develop diastolic dysfunction, systolic dysfunction, even in basal condition. So we believe that this pathway is important. And in this case, AKT activation, GSK-3 beta inactivation is directly involved in disease. And this is most recent conclusion. So then how to apply this knowledge to clinical condition? In the previous concept is that when AKT is activated, FOXO is phosphorylated to 3 sites, and when AKT is activated, FOXO exit from the nucleus. So no chance to reactivate the FOXO. But our data suggests that this actually downstream mechanism is more important for the nuclear translocation of FOXO. And in fact, that today I do not show the data, but if I use the FOXO 465, 467 D mutant, also mimicking mutant, even when AKT is activated, FOXO can go into the nucleus. So that rather than conducting modulation upstream mechanism, downstream mechanism, including GSK or even FOXO or even FOXO deacetylation is a valid way to mitigate the Western diet induced cardiomyopathy. And as you know, NIH want us to study humans, and I recently started the collaboration with Susumita Safu in New York, and she recently started the collection of human heart who suffered from obesity or type 2 diabetes. And she collect a fresh heart sample from those patients when those patients die. And she very kindly shared her sample with us. So we got the sample control heart, obesity heart, type 2 diabetes heart. And very interestingly, these disease heart all increases GSK phosphorylation. So this is similar to Western diet, interestingly, rather than high-fat diet. And we saw ATG level goes down. So this clearly indicates that studying Western diet and ATGL, AKT, GSK connection is important. So to summarize my talk, GSK activation is compensatory during high-fat diet induced cardiomyopathy, and compensatory mechanism is mediated by ATGL upregulation, and this certainly prevent lipotoxicity. And the mechanism is the FOXO1 phosphorylation, and this actually allows nuclear translocation and the interaction with SAT1 and SAT1 FOXO1 deacetylation, ATGL upregulation. But more importantly, I found that this pathway is inactivated with Western diet, and this downregulation plays a pathological role. So past the next 5 minutes or so, I would like to discuss several things. And although I said that ATGL upregulation is a good thing, but I know that Gary Ropaschuk and others, uh, overactivation of fatty acid is certainly increases oxidative stress. So we really need to find out the uh, optimum range of the ATGL. And also, FOXO1 has many functions. And some people understand that the FOXO1 is a very bad guy for the heart because it activates apoptosis. But some people say it's a good thing. And my understanding is that it is very important for upregulation of antioxidant gene and autophagy. So how do we explain this? And we believe that post-translational modification is really important. And the most well-known mechanism is AKT-mediated phosphorylation, and that this induces nuclear exit. But we recently found that Hippo pathway MST1 phosphorylate heavily at the DNA binding domain of FOXO1, and this actually increases nuclear translocation. But since these actually become acidic, and despite the fact that the FOXO goes into the nucleus, it release nuclear DNA binding is decreased, but FOXO1 start to bind to CBP beta and activate antioxidant. And today I show that 465, 467 induces nuclear translocation. And also it has been shown that FOXO deacetylation induces nuclear translocation. So this indicates that when we study FOXO1, making simple gain or loss of function doesn't may not be a right way to assess the FOXO function, and we really have to look into how FOXO function is mediated by post-translational modifications. And also, we are so curious why high-fat diet and Western diet differentially affect this signaling pathway, and these are unsolved question. And finally, 2-3 minutes, our lab has been interested in GSK-3 beta function, and if we mutate serine 9 residue to alanine, this make GSK-3 beta constitutive active. But nowadays, we have to study the cell type effect specific role of a GSK-3 beta. So we decided to make a conditional this knock-in, and I have a good collaborator to design CRISPR-Cas9 mediated gene modification, and he basically inserted uh, uh, two sequence, exon 1B sequence. This is a wild type and flipped sequence with a mutation. And when Cree excised this site, this reinserted so that this mutation can be expressed in Cree-dependent manner. So this actually is a very, very useful model. And it turned out that GSK's function is very much cell type specific. And we used to have this constitutive active knock-in mutant. And 2009, we report PNS that if you knock in constitutive active, cardiac function improves. But if we use cardiac specific knock-in, ejection fraction is rather decreased. And we are so upset that systemic and cardiac specific result is opposite. But if we make a fibroblast specific knock-in, we saw the same thing as systemic. So in terms of pressure overload, GSK3 function, I think, fibroblast effect is predominant. But as you see here, if we use the cardiac specific knock-in for Western diet and pressure overload, certainly Western diet, lipotoxicity and diastolic dysfunction is improved. So this actually S9A knock-in in cardiomyocyte is certainly protective. But the pressure overload, as I told you, that the more fibrosis, observed. So certainly the effect is not only cell type specific, but also stimulus specific. And so that's actually make the GSK treatment using GSK is difficult. So this is a final slide. And I saw many things about the molecular mechanism starting from AKT, GSK, FOXO, phosphorylation, deacetylation, SAT1s. And so you may ask, so how do you want to utilize this knowledge for clinical treatment? And my opinion is very difficult, but certainly upstream mechanism has more different downstream effect. AKT not only affect the GSK but other molecules. GSK also affect the other molecules. But if you go really downstream mechanism such as phosphorylation of these two sites or FOXO1 deacetylation, we may be able to find a way to upregulate IDGL to prevent the lipotoxic conditions. So with that, I would like to finish my talk by thanking my fellows. And especially today's work is done by Koichiro Takayama and also his successor, Takuma Takada. And thank you very much for your attention.
Thank you, Junichi. We're grateful to you for your fantastic presentation and congratulations for defining a new pathway for modulating obesity. With that, I would like to open the presentation to panel discussion and question from the audience. Meanwhile, I see Dr. Buddha Don. He's the president of IACS. I'd like to acknowledge him as well. Thank you, Buddha, for joining us. All right. I see Professor Jason Dyke. Your hand is up, at least digital hand. And
Yes.
then Professor Tagmeyer.
Right. Wonderful work. Very exciting. I mean, we spent a number of years just manipulating ATGL, and it's nice to see you going upstream
Thank
and
you very
finding out what actually is responsible for that. So it's really, really fantastic work.
Thank you.
I just had a few questions about, well, first of all, one of them would be on the difference between the Western diet and the high-fat diet. I'm assuming you performed glucose tolerance tests and measured insulin levels. Like, I'm guessing one is more severe than the other.
Yeah, that's an experiment to be done, and I'm sorry that this relatively new direction. So we really have to. I certainly write down, important things to do. Yes.
Yeah, and so the other thing I was wondering about the time course of your experiments. I know with and maybe Heinrich will agree or disagree, but I think lipotoxicity, when you look at ATGL, especially when you're looking at, I think lipotoxicity takes two forms. When we knock out, with Rudy Zechner, when he knocked out ATGL in the cardiomyocyte, you get this very fat, almost white heart. And in itself, that high levels of lipid accumulation is quote unquote toxic in terms of just, I think it mechanically restrains or constricts the heart. So there's a lipotoxicity there that is due to too much lipids, and then conversely, by driving it out of the TG pool with ATGL, you're potentially, in some ways, as everyone knows, the triglyceride pool is inert and fairly happy place for fats to sit. When you drive them out, then they have the potential to become toxic. So I'm wondering if you're looking at a balance, it's like the Goldilocks kind of, say too much of ATGL is good or not good, and too little is not good, and you need somewhere in between. So I wonder what your thoughts are about that.
Yeah, I think that's a extremely important question, and we have done some lipidomics analysis, but we haven't extended study to the genetically altered mouse model that I presented today, and these are the extremely important questions that need to be done.
Yeah.
Very good. With that, let's move on to Dr. Teigmeyer and then Dr. Penani. Dr. Teigmeyer.
Yeah, thank you very much, June, for excellent presentation and eye-opener. Moving down to the molecular mechanisms, we in the past have taken a bit more of an outsider's approach by using the isolated working height and the rates of substrate utilization. And 2007, we found the same striking difference between Western diet and high-fat diet. And high-fat diet, it seems the heart seemed to have adapted, as also supported by an upregulation of long-chain fatty acid and oxidation enzyme activities, and even mitochondrial mass. And the Western diet was detrimental. And now, it appears to be the molecular mechanism. And we used the term glucolipotoxicity to reflect both of those components. And we got a bit into hot water with our diabetes colleagues who at the same time actually found a similar phenomenon in beta cells and beta cell dysfunction. And so we tried to play it down a little bit, but what it is the principle is very interesting. There is something like metabolic stress and fuel overload, and it raises the question whether insulin resistance is actually initially an adaptive mechanism preventing the fuel overload. What do you think about that idea? And then another interesting phenomenon that we've become very interested in recently is in aging, the phenomenon of sarcopenic obesity. So the paradox of catabolic, anabolic metabolism simultaneously.
Yeah. So
the fuel toxicity hypothesis and anything that couples fuel uptake and oxidation should be beneficial to the heart. And that's, I think, what your work actually demonstrates indirectly, but very nicely.
Yeah. So actually, myself, I'm not obese, but I develop glucose intolerance and other things, and so I'm very interested in what's the underlying mechanism, but I don't have animal model that properly address this, and certainly Dr. Tegmeyer raised the issue is important, but I do not have a personal data to say something about it.
Thank you, June. Let's see. We have next was I think Dr. Penani, Professor Francesco.
Thank you. Congrats, June. It was really a fantastic set of data, and congrats for delineating this new pathway. I have a question about, going really downstream to the path and moving towards chromatin. I was actually thinking what's going on at the level of, chromatin accessibility at the promoter of ATGL. Have you checked whether, FOXO can find an hostile environment, a very repressive chromatin or a more accessible chromatin, whether this could be part of the game in the regulation of ATGL transcription?
So certainly the acetylation, this is transcription factor acetylation, but the third one causes histone deacetylation, right? So I think not only transcription factor but also chromatin accessibility is affected, and also we have data that recently submitted to cardiovascular research, and the third one actually compete with a core transcription factor complex, thereby affecting POL2 recruitment. So there are many layers of regulation, and but in terms of histone modification, chromatin effect, that's actually the possibility, and we really want to find out what's going on there.
Yeah, I mean, this could also explain why, I mean, with different stressors, you see also different things, not only among metabolic stressors, but also when you compare, a mechanical stress with a metabolic one. So I think chromatin could be part of this pathway. Quickly, a follow-up questions. I have to ask you something about autophagy. So have you checked what happened to autophagic flux when you modulate the TGL? Do you think that autophagy can have a causal role at the end in the cardiac phenotype upon a TGL manipulation?
So I haven't done ATGL modification, but Ron Tian's group has shown that fatty acid oxidation directly regulate autophagy levels. So I think certainly ATGL would affect autophagy, but we haven't done any on that.
Thank you.
Thank you, June. I have Christoph Mack next on the line for the question. Then we go to attendees. Christoph.
Yeah, June, thank you very much for this really very clear and nice talk. And
thank you.
yeah, I think it's remarkable. I mean, also alluding to what Heinrich said, that the different diets induce different effects on this pathway. So sugar seems to be worse than fat. And I mean, as you know, I'm working also a little bit more downstream on the mechanoenergetics and EC coupling. And indeed, we also observed in rats that a high-fat diet alone it did not harm EC coupling much. The cells after 16 weeks of high-fat diet were happily contracting. The mitochondria were fit. They were even slightly better than the normal treated ones. So fat alone seems to be okay. So it's interesting now to find out what the sugar adds to it. But maybe when speaking of combining stressors, and also Francesco mentioned this. So I mean, the HFpEF model many people use is a combination of high-fat diet and L-NAME. Okay, L-NAME, high-fat diet, okay, it's fat eating, that's quite natural, but L-NAME, of course, it can be a little bit artificial, but it recapitulates nicely the HFpEF phenotype. And I think it's the concept of combining the diet component and the mechanical component, as Francesco also mentioned. So maybe I missed it, but have you looked at in these conditions a little bit as well?
Oh, so we have previously shown that if you give pressure, we also use a HFpEF, the Joe Hill's model, but also a high-fat diet plus a pressure overload, and that actually causes totally different, signaling pathway, and actually YAP is upregulated and myocyte dedifferentiated. So I completely agree that high fat plus some mechanical, additional stress causes activation of a harmful mechanism.
And you mentioned at some point the potential downstream consequences, if I may ask another question, such as apoptosis and antioxidative genes regulated by FOXO and autophagy. So I was always, I'm always a little bit puzzled how fat causes oxidative stress. Does your mechanism, can it explain it? Is it through regulation of antioxidative enzymes? Because it's otherwise for me sometimes a bit difficult to understand how fat causes oxidative stress.
Yeah, the FOXO1 is a very important transcription factor for the SOD and catalase, and when it binds to it comes to the nucleus, and we also believe that additional transcription factor is needed to interact, but antioxidant is so without the FOXO mechanism, certainly oxidative stress is increased.
Okay, thank you.
Again, let's see. We have one of the participant has a question. Forgive me if I don't say your name right. Dr. Thomas Pulinil Quinel. Do you, I guess, Natalie, can we activate Dr. Thomas's microphone, or you have written as a chat box? No, there's nothing in the chat box. So
can you hear me? Can you hear me?
Okay,
yes,
yeah. Go ahead.
Thank you for that. Two quick questions. First of all, the talk was great. Thank you so much, Dr. Sadoshima. My first question was, in your models, what happens to PPAR? Because PPAR also encodes ATGL and many other genes. So what happens to PPARs, especially PPAR alpha in your model? That was my first question. And my second question was, atiglastatin, which is an ATGL inhibitor, has shown to be cardioprotective in certain conditions and where the ATGL is inhibited. But in the current model that you show, that compensation is important and it's going down is considered bad. So I was wondering, how do you reconcile these two pathways, wherein one pathway of ATGL inhibition protects the heart, but in your model it's the opposite? So just to get some clarity on it.
Yes. So the ATGL, the hydrolyzed and the free fatty acid is formed, and the PPAR is certainly activated, right? So I think when ATGL is upregulated, high fat diet case, we have a data that the PPAR target is upregulated. So we have not done whether this mechanism is inhibited when ATGL is downregulated or GSK-3 is inhibited. So it's a good question, but my prediction is that ATGL should not only helpful for making ATP, but PPAR transcription is also important. And ATGL inhibitor, yeah, certainly some condition ATGL is overactivated, and but we need to find out when it happens. And I'm not surprised that in some condition ATGL inhibitor is protective. So that's my opinion.
Thank you, June. I have Lori had a question. Dr. Lori Kirschenbaum. His microphone is off. All right, Lori, turn on your microphone, please.
Oh, yes. Thank you. I'm sorry my camera keeps glitching in and out. First off, June, congratulations. It's really wonderful to see the story unfold like that, and it's really great to see everyone else, even though we were maybe together less than 24 hours together on the other side of the world. It always gets to me how you can be on one side of the world, and a few hours later you're somewhere else. June, I wanted to ask you about the perlipins and lipophagy. So, Anna Maria Corvo had published a paper several years ago that the perlipins were needed to deprotonate the lipid droplet for ATGL to get in. And this was a chaperone-mediated process. So have you looked at the perlipins in this case, and are they
So this is the question often asked, I'm asked, but basically lipophagy is a lysosomal dependent degradation, right? So I worked so hard, but so far I was not able to observe a clear image of autophagosome containing lipid droplet. So maybe I should create extreme conditions such as high-fat diet plus lysosomal inhibition or something like that, so that
Okay.
stays. But I do not know the contribution of that mechanism in my mind.
I was going to ask you, and maybe Jason or the others who are more expert than me on the lipid droplet synthesis, maybe even Thomas, the question about the lipid droplet size. Many times we see, and I saw the talk the other day, the lipid droplet numbers will increase, or sometimes the size of the lipid droplet increases. And I'm wondering if this is just an amalgamation of small lipid droplets that are coalescing to make bigger ones, or is it just, they're accumulating, they're not being metabolized? So I don't know, June, if you want to comment on number or size of lipid droplet, or anyone else maybe want to drop in and help me out here, what that really represents.
So in my study, I saw increases in number, but when I studied the ATG7 knockout mice, I saw enlargement of lipid droplet. So how this size is regulated, I don't know. But
Jason, any thoughts?
I think both happen, to be honest. I think what you're seeing is in certain situations, you're getting, I think, a fusion of these lipid droplets. And then also, I think the localization of it is can drive, like where they're located can drive whether they have the spatial capacity to get larger as opposed to just have more of them. So I think it's a non-answer, I guess, but bottom line is both can happen.
Yeah, the reason I ask is because in some cases, if you have an autophagosome that's too big, whatever too big means, it's difficult for it to be digested. And I was thinking if there was lipophagy going on specifically, although June mentions he didn't see it, that maybe these lipid droplets are so big or whatever, and they can't actively be digested. But it was just something I was thinking about. But thanks very much for the answers.
Very good. We have one more question from Alan Titus. Forgive me if it's Titus or Titus. And we can activate your microphone so you can ask your question. Professor Natalie, our capable coordinator, would you activate Alan's microphone?
Yes, just did.
Yes, can you hear me?
yes, I can hear you, Alan.
Yes, Dr. Sadoshima, I'm happy to see you here. And also, congrats on completing that story which Koichiro started. So my question is with regards to the chromosome accessibility, which was asked by Dr. Mac. So when we look at fat as a source versus carbohydrate as a source, do you think that differences in that source, for utilization for metabolism, that can have some epigenetic modification, because fat has more acetylation epigenetic marks, whereas carbohydrate sources can cause more methylation changes in epigenetics, and then the chromosome opening and heterochromatinization can vary. So do you think that could be a reason why we see that fat versus that Western diet has any changes in the phenotype we observe?
Yeah, so thank you for the question, Alan. Yeah, nowadays, the metabolites directly affects various mechanism, histone modification, right? So acetylation is certainly a possibility, and so, but this is certainly a future direction of the research.
Excellent. I see one more question, last question. Dr. Festina Cucci. You activate your microphone so you can speak. Good.
Yes, thank you. Thank you very much for your talk. I was wondering, have you thought to involve also mitochondrial beta oxidation or CPT1 or PGC1 alpha, knowing that you found a mechanism that is correlating due to SIRT1 and FOXO1? I heard that someone mentioned lysosomes. I agree also with the lysosomes, but also mitochondrial involved in lipid accumulation could be one of the mechanisms that you can think to analyze.
Yeah, so certainly oxidative phosphorylation and also PPAR signaling affects oxidative stress, also affects mitochondrial function. So everything is connected, but I really have to check whether which mechanism is affected by genetically altered mouse models. Thank you for the good question. Thank you.
All right, I will go ahead and acknowledge everyone on the screen. And from the panelists, first I see Junichi Sadoshima. We're truly grateful to you for your fantastic presentation. Despite being sleep deprived from overnight flight and also staying late answering all the questions, our sincere gratitude to you. Next, I see Lori Kirschenbaum. Lori, thank you again for participating. Then I have Gaetano Santulli. We're grateful to you as participating as a panelist. Jason Dyke, thank you again. Our gratitude. Distinguished Professor Tagmeyer, we're really honored to have you